Hi, my name is Art Arnold, and I'm in the Department of Integrative Biology and Physiology at UCLA. Today, I'm going to talk about sex chromosome factors that contribute to sex differences in disease. So there are really 4 points in today's lecture. First of all, in mammals, there's an inherent sexual inequality in the expression of X and Y genes in every tissue of the body, and these cause sex differences in these different tissues. But we've not known what are the genes on the X and the Y chromosome that cause these effects by and large, especially outside the gonads. We approached the study of this question through the use of two animal models, two mouse models, the 4 core genotypes and the XY star model. And these studies have led in the last few years to the identification of 2 X genes, Kadium6A and Kadium5C, and a Y-linked gene, UTY, as primary agents causing sex differences in different tissues. Our approach to the study of sex differences really stems from a theoretical viewpoint. The old theory from the last century was that the inherent sex difference in the X and the Y sex chromosomes plays out first in the gonads. Individuals that have a Y chromosome and the SRY gene develop testes, and those without the Y chromosome develop ovaries. And so the sex chromosomes determine the kind of gonad, and that's the first step of sexual differentiation called sex determination, and it's a genetic step. The second step is called sexual differentiation, and it's hormonal because the gonads secrete different hormones, either testicular hormones or ovarian hormones. And these act throughout the body to cause sex differences in traits. This model is in the textbooks and is still taught today. A lot of people still think this way. But the model is inaccurate because sex differences develop before the gonads do. And they develop after the gonads do for reasons that have nothing to do with the secretions of the gonads. So we proposed another idea, another model, which is that the first and foremost sex difference, which is genetic, which happens in the zygote, the individual is either XX or XY. That plays out to cause gonadal differentiation as in the old model, and hormonal effects throughout the body, which are quite potent. But in addition, these differences in the sex chromosomes play out in other tissues of the body, and these are called sex chromosome effects, and they cause sex differences in physiology and disease as well. But we've known about the hormonal effects really for 60 to 100 years, and so the hormones have been well studied, and we know what they are. The different testicular and ovarian secretions that make sex differences happen elsewhere are testosterone, estradiol, progesterone, and anti-Müllerian hormone. But we don't know which are the genes on the sex, what are the primary agents of sexual differentiation coming from the sex chromosome via the sex chromosome effects. These really haven't been known, but as I'll mention, they've been recently identified, at least the first few. We don't know them all. We have known for some years what classes of genes on the sex chromosomes might be causing sex differences. And I've written about 4 classes. There might be 5 or 6 or 7 different classes, but the primary classes are, number 1, Y genes, which are present only in XY cells, will cause XY cells to be different from XX cells. The second category is the dosage of X genes, that having 2 X chromosomes inherently makes X dosage higher in those cells than in XY cells. But this X dosage difference, of course, is mitigated, or for most X genes, 1 X chromosome is turned off in the XX cells by X inactivation. And so the dose of X genes is made more equivalent in XX and XY cells. Now, there's a whole class of genes that escape X inactivation and therefore still are expressed higher in XX than XY, and we'll be emphasizing those today. A third class that we'll mention in passing is that the parental imprint on X genes differs in XX and XY cells because XX cells receive both a maternal and a paternal imprint, but XY cells only receive a maternal imprint. And I won't mention for this talk any other classes, of sex difference. So today we're going to emphasize this X dosage difference, that some X genes are expressed higher than in XX and XY cells. The model that we begin with for studying sex chromosome effects is called the 4-core genotypes model, developed by Paul Burgoyne and Robin Lovell-Badge at the National Institute for Medical Research in London. And this model is particularly good because you can compare XX and XY mice that have the same kind of gonad as we see in the next couple of slides. A typical male on the left has the SRY gene, the testis determining gene on the Y chromosome, and that male makes two kinds of sperm, either Y-carrying or X-carrying sperm. Whereas in the 4-core genotypes model, the SRY is not on the Y chromosome. It's been lost through a spontaneous deletion, and the SRY gene is inserted as a transgene onto chromosome 3. So this animal can make X sperm either with SRY or without SRY, and similarly can make Y sperm with SRY or without SRY. Thinking that the red X chromosome comes from the mother here, the father then makes 4 kinds of babies. He makes XX babies that are normal females and have ovaries. He makes XY minus babies that don't have SRY and they have ovaries therefore too. And he makes XX and XY animals that have the SRY transgene and therefore they have testes, and they're called males. So this is really a 2-by-2 with half of the animals being XX and half of the animals being XY. Half of the animals having SRY and half of the animals without SRY. The animals with RY have testes and testicular secretions. The animals without SRY have ovaries and ovarian secretions. And so if we find that a sex difference in a trait goes along with whether the animals have testes or ovaries, so if the gonadal males differ from the gonadal females, then we say that the trait is caused by hormonal effects. But if, on the other hand, the XX animals are different from the XY animals, then we say it's a sex chromosome effect, not dependent on the gonads, not dependent on the type of gonadal secretions. So here's an experiment, a simple experiment of a sex difference. Male mice are larger than female mice. And we weigh the animals at day 21 and the day of weaning, and we're measuring body weight. And there are 4 groups. They are either XX or XY, and they have ovaries called gonadal females, or they have testes, the black bars, called gonadal males. And you can see at weaning there's not much difference among the 4 different groups. But if we wait 24 days thereafter and let puberty happen, the hormones kick in, and the animals with testes, the 2 male groups, weigh more than the animals with ovaries. And that suggests that body weight is predominantly caused by gonadal secretions. But in addition to that, the animals that are XX are slightly larger. There's a slight effect of sex chromosomes. The XX animals are slightly larger than XY. So assuming that the animals are, the sex difference is caused predominantly by hormones, we do a classic endocrine experiment, which is to take out the hormones to see if the sex difference goes away. This is a paper in collaboration between Lab and Karen Ruiz at UCLA. And the animals are weighed at 2.5 months of age, which is the 0 week of the experiment. And the animals that have testes weigh more than the animals with ovaries. After you remove their gonads on that day, 4 weeks later, the sex difference has disappeared, and showing that the body weight effect was, sex difference was caused predominantly by gonadal hormones. But then something unusual happens, which is that over a period of some months, the animals that have 2 X chromosomes are bigger and fatter than the animals with that are XY. If you put them on a high-fat diet, the livers become very fatty, except that happens mostly in the XX animals, not in the XY animals. So body weight and body fat here is a sex chromosome effect in the absence of the gonads. Summary here is that with the gonads intact, animals with testes, called males, are larger. So the 2 blue animals are larger than the 2 red animals, the red animals being the animals with ovaries. But in addition to that, the XX animals are slightly larger than XY. If you take out the gonads, then the two XX animals become much larger and fatter than the animals that are XY. So then we use a second model, which was discovered by Eva Eicher at the Jackson Laboratories. And this is a model that has 4 different genotypes shown here as XX, XXY, XY, and XO. And they either have 2 X chromosomes, or they have 1 X chromosome, or they have a Y chromosome or not, but the number of X and Y chromosomes are relatively independent. So we do the same experiment at 2.5 months of age. We weigh the animals before we take out the gonads, and the 2 male groups weigh more than the 2 female groups as before. A month after taking out the gonads, the sex difference goes away, confirming the hormonal nature. But then over a period of months, animals with 2 X chromosomes, whether they have a Y chromosome or not, are heavier and fatter than the animals with 1 X chromosome, showing that the sex chromosome effect is an X effect. And so now we're getting somewhere as we're trying to narrow down where the genes are that are causing these effects. We know it's an X chromosome effect. These models now have been applied in numerous models of disease. And there are many diseases that are influenced by sex chromosome effects, showing that the sex differences in these diseases are in part caused by sex chromosomes, not by gonadal hormones. Now, I shouldn't say not by gonadal hormones. In most or all cases, there's also a gonadal hormone effect. So the sex difference is a mixture of hormonal and sex chromosome effect, and that's the typical case. And so there are numerous diseases that have been studied in many different laboratories. And so far, there's been about 80 papers published. And then using the XY star or other models, we can ask, well, of these sex chromosome effects, how many are caused by the X chromosome? And here's a list of 12. And this is, in fact, not an exhaustive list, but many of the sex chromosome effects are actually caused by the number of X chromosomes, not by the number of Y chromosomes. So now we would like to know, okay, precisely what are the genes causing these sex chromosome effects? And as I've said before, we know what hormones are the primary agents of sexual differentiation causing the hormonal effect. But now we want to find the new testosterone, the new agents in the sex chromosome category that are causing these effects. Now, X inactivation is actually a process that makes the 2 sexes more equal, because females have 2 X chromosomes and males have 1 X chromosome. By shutting off, transcriptionally silencing 1 X chromosome, the X inactivation process makes females have 1 active X chromosome in each cell, each somatic cell, just like an XY cell. So XX and XY cells generally have 1 active X chromosome. But then many genes can escape this X inactivation process. This is a study by Tukiainen and others, mapping the human X chromosome for sexual bias. And these are different tissues from the GTEx consortium, and different X genes mapped along the X chromosome here. And so each column is 1 gene expressed in different tissues, and the red squares are the genes that are expressed higher in XX than XY. The blue squares are genes higher XY than XX. And you can see that there are quite a few genes that are sexually unequal in their expression. And there are, on the non-pseudo-autosomal part of the X chromosome, there are genes that escape X inactivation, and there are about 25% of the genes, or 23%, are expressed higher in XX than XY. That's the second class of genes that cause sex differences that I mentioned earlier. And so, this slide shows that 4 genes in mice that are some of the main genes escaping X inactivation. They're encoded by the X chromosome. And here are 2 genes, KDM6A and KDM5C, which are histone demethylases. And you can see that in the 4 core genotypes, the 2 XX animals, either males or females, express Kadium-6A higher than the 2 XY animals, and this is in, I believe, in adipose or liver, I forget which. And these escapees have this pattern where they're higher in XX than XY and can therefore lead to sex differences. Our experiment to prove which of the X genes are causing the sex difference in adipose or other tissues is to take an XX female and then knock out one copy of the X escapee in one group of animals. So one group of animals is plus minus for the gene of interest, let's say Kadium-5C, and one group is a wild type and has 2 copies of Kadium-5C. And so now we're varying Kadium-5C 1 versus 2 copies instead of varying 1 versus 2 copies of the X chromosome. And that's schematically shown here, and we can compare the protein levels to confirm that in fact the knockout works. Okay, when you reduce the dose of Kadium-5C from 2 to 1, the body weight of the animals goes down just as if you were comparing XX and XY or XX and XO. That is, the dose of Kadium-5C explains much of the sex chromosome effect. And this then identifies Kadium-5C as an agent of sexual differentiation because of its inherent sex difference in dosage. This is also true for body fat animals with 2 versus 1 copy of Kadium-5C here show differences in body fat, and it doesn't matter whether they're on a chow diet or a high-fat diet, you get the same dosage effect of Kadium-5C. So our pathway to discovery of the sex chromosome effects was, first of all, to use the 4 core genotypes model to find that XX and XY animals were different even if they had the same kind of gonad. Then to compare XX and XO animals to find an X dosage effect or an X chromosome effect. And then to manipulate the level of one specific gene all by itself, keeping all the other factors constant. So these experiments have been performed several times over. The example I've been using was done in Karen Rui's lab in collaboration with us, and showing a dosage effect of the histone demethylase, Kadium-5C, causing differences in metabolism and in differentiation of adipose cells. In other labs, similar kinds of experiments have been done. In the lab of Rhonda Voskuil at UCLA, they study experimental autoimmune encephalomyelitis, a model of multiple sclerosis, in which a mouse is injected with an autoantigen, a myelin protein mix, and an immune reaction is induced, and then the animal shows a multiple sclerosis-like disease. So over time, the animal is injected on day 0, and then the clinical score here is mapped, in which a healthy animal gets a very low score, and an unhealthy or animal that has difficulty walking and righting itself gets a high clinical score. And these are comparing animals that both have testes. This is the 4 core genotypes model, and the XX animals do worse and stay worse than the XX animals. So in this case, being XX makes the animal worse than being XY, and you can see different levels of inflammation also caused by sex chromosome complement. Then the Voskuil group knocked down another X escapee, Kadium-6A, in CD4 positive T cells, and showed that this changed the disease profile as well. So the sex chromosome effect is explained in part by the dosage of Kadium-6A in T cells using, this was a conditional knockout using a Krelok system. So here's the same kind of clinical experiment. The animal is injected with the autoantigen early in the experiment. And here the clinical score goes from healthy to unhealthy. The wild type animal gets sick and stays sick, the disease with a high disease level, whereas the conditional knockout of KDM6A in the T cells protects the animal. So in this case, a high dose of KDM6A is harmful, and a low dose of KDM6A is protective in the disease within the T cells specifically. A third example of this kind of approach was done in the laboratory of Dina Dubal at UCSF, investigating Alzheimer's disease. So in Alzheimer's disease, the male mortality is greater than the female mortality in human populations. And this lab uses a model of Alzheimer's in which a humanized amyloid precursor protein is inserted into the genome of the mouse. And in wild-type mice, you can see on the left side here that if you're comparing two animals that have the humanized gene inserted into their genome, and this kills the animals prematurely, and the females survive better than the males in wild-type animals, X females versus XY males. In the four core genotypes, the animals with two X chromosomes, whether they are males, gonadal males or gonadal females, do better than animals that are XY, showing the sex chromosome effect. And in the XY star model, where you're independently manipulating the number of X and Y chromosomes, animals with two X chromosomes, either XX or XY, do better than animals with one X chromosome, showing that it's an X dosage effect. They then manipulated, again, the KDM6A gene, which escapes X activation and is expressed higher in XX than XY. So in one experiment, they manipulated the levels of KDM6A in an in vitro experiment by knocking down KDM6A with interfering RNA. The KDM6A here is protective, so the knockdown, these animals are the knockdown group, have low levels of KDM6A, and they have higher levels of toxicity to the A beta molecule when KDM6A levels are low, whereas the wild type animal that gets scrambled, interfering RNAs, has less neurotoxicity, showing a protective effect of KDM6A. In another experiment, the Dubal group overexpressed KDM6A in the hippocampus using a lentivirus and injected into the hippocampus of live animals. And they show that this then increases the level of expression of KDM6A in the hippocampus. And in a learning paradigm, the animals that are carrying the Alzheimer's allele, the humanized APP allele, the XY animals, which normally have low levels of KDM6A, if you increase the KDM6A, you increase their ability to learn. And that shows a protective effect of the KDM6A in an animal that normally has a low level of expression. Another example is a KDM6A effect on bladder cancer in the laboratory of Sean Lee, who was at this time at Harvard. And in this case, a drug is injected into animals that induces bladder cancer and reduces survival. And in the four core genotypes experiment, animals, the female groups, that is the animals with ovaries, live longer than the male groups, showing a hormonal effect. But in addition, in both sexes, the XX animals do better than the XY animals, and they live longer, showing a sex chromosome effect. And when they knock down KDM6A, so here's the animal, an XX animal that has normal levels of KDM6A and is wild type. If they eliminated KDM6A, which is normally higher in XX than XY, then the animals did not live as long, showing that the KDM6A was protective in this case. And the final example comes from, it's actually a Y chromosome effect, not an X chromosome effect. And it's from the lab of Mansoura Eghbali at UCLA. And they study pulmonary hypertension and other cardiovascular diseases. Pulmonary hypertension affects women much more than men. It's untreatable and it's often a fatal disease. And this pulmonary hypertension can be induced in mice by putting them in a hypoxic chamber in a low oxygen level for several weeks. And at the end of several weeks, you can measure, among other things, the pressure in the right ventricle. So in the 4 core genotypes experiment, the animals with 2 X chromosomes have higher pressure than the animals with X and Y chromosome, showing that XY is protected in some way. To determine whether this is an X or a Y effect, they used the XY star model. And here this shows that animals with a Y chromosome, either XXY or XY, do better, have lower pressures than animals that don't have a Y chromosome, either XX or XO, showing this an X chromosome, a Y chromosome effect. And subsequent experiments have led to the discovery that it's the gene UTY on the Y chromosome that is protecting males in this mouse model. So our pathway to discovering new agents of sexual differentiation has been to study the inherent inequality of X and XY cells, starting out with the 4 core genotypes in the XY star models. And noting that both sex chromosome and hormonal effects usually operate together on the same traits, but their interactions are not well studied. And recent studies have identified and discovered new agents of sexual differentiation, K6A, K5C, and UTY. And this now opens up the study of mechanisms downstream from the action of these genes. So these experiments have been highly collaborative, and here's a list of some of our collaborators at UCLA. And of course, we got the mice originally and learned from Paul Burgoyne, who sadly passed away last year. And both Paul and Robin Lovell-Badge have been tremendous collaborators and colleagues. And we thank the NIH and other agencies for support. Thank you very much.